Hey everyone, I'm here in the Human Physiology Lab, and today we're gonna do the diffusion lab. I'm gonna show you the procedures and then we're gonna look at the results. So let's get started. All right, the first thing we need to do is take our dialysis tubing and get it wet so that we can open it up. So I'm just gonna pour some water into this beaker and let the dialysis tubing soak for a while. All right, it's been a couple of minutes, so the bag is ready now to be opened. So I'm gonna take it out of the water. And on one end, I'm gonna fold it back on itself and then clamp it off so that we don't have any leaking. Like that. And then on the other end, I'm just gonna rub it between my fingers to open it up. Okay, so the bag is now open. All the way down. Okay, so next up, I'm gonna get a pipette, a glass pipette, put the bulb on it, and I'm gonna put in about 4 pipettefuls of the 30% glucose. Okay, next up, I'm gonna add about the same amount of starch. I'm gonna use a different pipette so we don't get contamination of our solutions. This is a solution of 2% starch, and I need to shake it up first because sometimes the starch will precipitate at the bottom. So I wanna make sure it's all mixed up. So I'll add about 5 pipettefuls of the starch. Okay, so then we can kind of roll it around like that to mix up the glucose and the starch solutions. Okay, I'm gonna rinse this off to make sure there's no starch or glucose on the outside, and then I'm gonna add it into the beaker. Okay, so now we have our bag filled with starch and glucose. Take a look at the color here and write it down as the initial color in the bag in your table. Okay, so then to our beaker of water, I am going to add the iodine potassium iodide, so this yellow stuff here. I'm just gonna dump the whole thing into the water so it's nice and yellow. Okay, so inside of the beaker we have the water and the I2Ki. And then I'm just gonna add my bag to that, other side, to that. Make sure it's totally submerged in there. Okay, and now we just wait 30 to 45 minutes for diffusion to take place between the bag and the beaker solution. Okay, it's been about 30 minutes. Let's take a look at our beaker and bag. So this is the bag. Okay, so make note of that color. Write it down in your table. Okay, so that's the bag after 30 minutes. And then this is the beaker after 30 minutes, so make note of that color as well. Okay, so that should give you some information about the I2Ki and the starch. Now we need to do a test to see what happened to the glucose. Is the glucose still in the bag, or did some of it come out? And so we're gonna run a Benedict's test, and I have 3 tubes here. So one is labeled beaker. I'm gonna put some beaker solution in there. And another is labeled bag, and so I'm gonna put solution from the bag into this one. And then the one labeled control, I'm just gonna put water into there. Okay, so let's go ahead and do that. Okay, so we have about the same amount of liquid in all 3 tubes. And now we need to add the Benedict's reagent. So that's this thing in blue. And we're going to try and add about an equal amount to each tube. So I'm going to do about 1 pipette full for each one of those tubes. Okay, so let's look at the color before we heat these up. Okay, so this is the control, so make note of that color. This is the bag. And this is the beaker. Okay? All right, so I'm gonna add them to this water bath and heat them up for about 5 minutes, and then we'll take a look at the color. All right, so it's been about 5 minutes, and these tubes have been sitting in boiling water. So that gives the Benedict's reagent enough time to react with glucose if it's present. So let's take a look at the colors. So this is our control tube. Okay, so make note of that color after 5 minutes. Okay. This is the bag. Okay, so this solution has the liquid from the bag, the dialysis bag, plus Benedict's solution in it. This is after 5 minutes. Okay, so make note of that color. Okay, and then this is the solution from the beaker, right? So I took out the liquid from the beaker after it had done its diffusion. I added in Benedict's solution, and then this is the color that it turned. Okay, so make note of that. Okay, so think about all the color changes that you've seen, and what does it mean in terms of starch diffusion and glucose diffusion and the I2Ki. Okay, so that's it. So now I'm just gonna clean up all of this. None of this is toxic, so it can just go in the sink and in the trash. And you should go ahead and analyze your data, think about what it means, do the worksheet, and then write your lab report. All right, hope that helped.